Hey, welcome back to Kitchen Science Biology Lab. This is take number 3 because I keep screwing up on a Sunday. So let's get back to science. So what have I been up to today and the last couple weeks? Well, one of the things I did today was I baked bread, which is why I'm getting so late and I lost my natural sunlight to do my video. But, you know, good crop food here with our wheat flour going into my bread. Last week, decided to get started on the gardening a little bit and grew some seedlings. So I germinated some corn coming up in there. I also germinated, there's a lot of seeds over there, beans. So these I think are either green beans or they're peas. I'm pretty sure this is the tray that I did the beans in. And last tray, because they're really cool looking as they're starting to germinate and come up out of the ground. Actually, these are the beans. Look at it more closely. Those other ones were the peas, but here are my little baby plants starting to grow after about a week in this pot in my somewhat sunny window growing these things. So why am I talking about crops? Well, today our lecture was all about biotechnology and genetically modified crops. So the wheat and the corn that I'm trying to grow over there, these are plants that since sort of the beginning of agriculture that we have been modifying as people through traditional breeding, taking the best of the best seeds, growing them the next year, taking the best of those seeds, growing them the next year, and doing it again and again and again until we get the crops that we want, that have a high yield and a good amount of food that we can eat that's nutritious and easy to grow for us. And so when we're looking at these crops that traditionally, you know, the ancestors of corn did not have that big grain, did not have a whole bunch of grains. So we worked on that over thousands of years to get it to be a good crop. During the 1900s, we discovered DNA. And with the discovery of DNA, many years later, we started developing biotechnology. So we were able to identify single genes, pull those out of the genome of the big plant, get that little individual gene, understand its function, and do research on it, and figure out how to take that particular gene and insert it into another plant. And this is what we do with genetic engineering or genetically modified organisms. So we take the gene out of one organism and we're going to insert it into hopefully a good spot that actually has some functionality in another genome. And in the lecture slides, there's there's a couple of examples of this. And you can also watch I think I put 2 videos in there that you can watch that also go over this. So a lot of crops that we eat every day without even knowing it are actually genetically modified. They're not required to label the packaging. If you shop at like an organic market, you'll see a lot more products that say non-GMO on the label on there. There's a lot of regulations as to what they can put on those labels of the food packaging for us to be buying it. So some of the examples of crops, my laptop's right here. So we have a lot of crops that are being bred and modified for insect resistance. So a lot of our corn and soybeans and even cotton for our clothing is genetically modified to include a gene from Bacillus thurinburgii, a little bacterial organism that this gene, once it's inserted into the plant, is still able to produce a toxin that kills the insects that are eating the plant. But this toxin is biodegrades prior to it getting to us for us to eat. So we have a lot of insect resistance, and again, I said like corn and cotton, soybeans, other insect resistance plants that have been bred and genetically modified are like tomatoes, potatoes. There's a lot of viral diseases that will kill off a plant that just basically kill the plant itself, and it's just a virus that's in the soil and in the air. We know a lot about those lately, don't we all? So in plants like squash, potatoes, and papaya, there are several varieties that are grown for this. Probably the largest category of genetically modified crops, though, are those that are bred for herbicide resistance. Roundup is a chemical compound that has an indole acetic acid plant hormone that causes the plant to sort of, when you spray Roundup on there, it kind of triggers the plant to just grow and grow and grow and grow uncontrollably and starve itself of its nutrients and kind of shuts down that pathway so it just doesn't function properly and just kills the plant. So we can spray this on a field to kill the weeds, but if we spray it on our crops, what's going to happen to our crops? They're going to die too. So we have made genetically modified plants that are resistant to spraying these different herbicides so we can kill off the weeds and not kill the plants. So there are a lot of different varieties of corn, rice, canola, so oil-based canola, sugar beets, which most of the sugar wheat comes from, flaxseed oils are really important, and also flax for linen. Cotton, radish, and soybeans are all plants that have been genetically modified for herbicide resistance. We also have plants that are modified for increased nutritional qualities, so like canola, soybean, and also rice. Also just the harvesting kind of categories, like when are these going to ripen? Does the grain shatter and fall off of the plant, or are we keeping it on there? And just sort of health characteristics of the plant. Wheat, tomato, cantaloupe, these are all things that are benefiting from these different GMO traits. So let's get down to the science lab today. A long, lengthy introduction there. But one of the things that we can do at home for kitchen science biology lab is that we're going to extract DNA today. And I can do this in my kitchen, which is pretty awesome. So the plant material that we're going to use today are strawberries. And the reason I'm using strawberries is, one, I had them in the freezer, and two, they're very mushy. I let them thaw. And so we're going to use our strawberries as our example plant. Other supplies we need: spoon, some water, another bowl, shaker of salt, some, dish soap, I'm going to show you the brand there, and some rubbing alcohol. All right, so first thing we need to do is we need to smash these up. So remember, plants have cell walls. If we just go ahead and mix all this stuff in there and just turn it gently, it's not going to break that cell wall. So we're going to mechanically separate these cell walls with a spoon. So first thing we're going to do is we're just going to smash and chop at it, turn it into like a strawberry smoothie. We could use a blender for this, but like I said, these strawberries are pretty soft. The fact that they were frozen is not going to interfere with our experiment. When I used to do research in the lab on plants, I could freeze the leaves of the plants and then later on extract them in a lab using more sophisticated techniques than a spoon. Used a drill and a little mortar and pestle. It was pretty fun. So we're going to smash it up so we get a strawberry goo. So we got our strawberry goo. So we have mechanically broken these cell walls in our strawberry now. And now what we need to do is we need to chemically break apart the cell membrane. So remember, cell membranes are made up of primarily phospholipids, so fats. And they also have some proteins. So we have fats and proteins, a little bit of carbohydrates, and maybe some sterols. So as we've been hearing in the news about washing our hands quite frequently, why are we doing this? Why are we washing our hands so frequently? To get rid of the virus and other particles on our skin. And that's because the soap breaks apart these phospholipid membranes. So they break apart fats and they break apart proteins. So I'm just going to add water. I'm going to add a 1/4 cup of water. Probably too much water, but to my strawberry mixture. One of the things we also need to add is our soap. So our soap is going to get in there and break apart our cell membrane, so just a small squirt of soap. And we also need to add salt. So if you remember our lab on tonicity, it's a little shake of salt there, that we were going to try and mimic the salt concentration inside our cells. There's definitely more scientific method than this. And now I'm just mixing it up and smashing it a little bit more. Got a big piece of strawberry there that's not quite smashed. So now we got, you can see it's just the strawberry. The water has turned pink because those are our cell contents. We have broken these cell walls. We're now breaking apart these cell membranes by adding the detergent. And now we're just going to mix it up a little bit. In a lab, when I was extracting DNA from plants, we would then put this into a water bath, a nice warm water bath, and let it sit there for a while to make sure that we were actually getting apart all of the cell membranes and getting the DNA out of there. So our goal today is we're going to see DNA from strawberries. All right, so I have an empty bowl, and what I'm going to very carefully and hopefully not make a huge mess and get it on the bread or the computer, because you know nobody wants salty soap water on bread, is I don't want to get this chunks of strawberry. I want to get rid of those. I just want the liquid. You know, if I had like a small sieve, or colander, I could use that, but I don't really have something that small here at home. So I'm just going to use the side of the spoon. I think one of you all, when we were talking in class, that you just use like the lid of a pan to drain off the water from your mac and cheese. Well, I'm doing that technique. It's not perfect. Oops, big chunk of strawberry just went in. Bunch just went on the table. All right, and I didn't bring a paper towel. I'll just leave that there for now. All right, I'm going to pick out some of these extra strawberry bits. I got a little bit in there. Maybe if I try to transfer some with a spoon. Even though it says take 3, I never got to this point before. All right, so we got it. This works. Just transferring a little bit of the liquid. Do not drink this because I put soap in it, right? I don't want to get sick from drinking soap. Okay, so now I got maybe... here about 1/8 a cup or 1/3 of a cup, and it leaked everywhere all over my table. So now what we're going to do is we are going to add alcohol. So the DNA molecule itself has a partial negative charge on it, a partial positive, red to blue. So partially charged molecule when we're looking at our DNA molecule. Oops. And so if we add alcohol to it, it's going to cause the DNA to precipitate, or instead of being in a liquid form, it's going to cause it to become solid. And so I'm just going to squirt in about half a volume here of what I got already in my bowl of alcohol. And something really cool happens immediately. I don't know if we're going to be able to see this, but immediately what happens is we start to see the DNA precipitating. So if I stir this around with my spoon, you can see swirls of white gelatinous stuff appearing in my bowl. And so now I'm going to use a wooden stick. It's actually a spatula handle. Again, using my kitchen, what I have at hand. And I'm going to stir this gently, because if we stir it too quickly, we're actually going to mix the DNA in. But if I pick it up, you can see the gelatinous snot that is DNA there. So DNA, when it precipitates out of the solution, stir slowly, is a clear liquid that has a gelatinous characteristic to it. And there's a lot in there. So even though, I didn't even pour all of my strawberry liquid in there, we have a lot of DNA. And so we can look at that. Hey, one of you just sent me an email. And play around with it, that this is millions of molecules of DNA precipitated out of just a strawberry dish soap saltwater solution, and we did it here at home. So I hope you enjoy my kitchen science lab. It's been a odd day, but here we are. And there you have it. If you want to fascinate your relatives at home because you're bored, grab any type of produce. Strawberries work really well. Onions work really well. So do apples. Other things you may have at home. Anything that you can smash really easily, like a peach, would also work pretty well. Strawberries are awesome because they are octoploid or hexaploid, so they have either 6 or 8 times the number of chromosomes, and so they have a lot of DNA. So hope you enjoy the lab. Hopefully there's some sort of scientific knowledge you gained today, and I look forward to hearing from you in the discussion. Now I have to go wash my hands.